Hey everyone, my name is Suliman Ayad. I am a PhD candidate in the Hansen Research Group, and today I'd like to share with you a video abstract from one of our more recently published papers entitled Enantioenrichment of racemic BINOL by way of excited state proton transfer. So first I'd like to talk to you a little bit about what excited state proton transfer is, or ESPT for short. So depicted here we have a naphthol molecule before and after excitation. And so once we excite the naphthol molecule, we have this electron density shift away from our acidic proton, which weakens the bond between the hydrogen and the oxygen there, which makes it much more acidic. pKa drops of up to 13 units have been observed for excited state proton transfer systems. Once this proton is now more acidic, it's much easier for a base to come along and abstract that proton. So some of the Hansen Research Group's earliest work with excited state proton transfer dyes was attempting to enantioselectively protonate this silyl enol ether group using bromobinol as depicted here on the left of the screen. Our hope was that using this atropisomeric compound would allow us to selectively protonate either the Re or the C face of our silyl enol ether here in order to achieve an enantiomeric excess. Unfortunately, the HPLC indicates that we had a nearly 50-50 mixture of the R and S enantiomers of our end product, despite having started from a completely enantiopure bromobinol. Later tests proved that bromovanol was capable of achieving a large enantiomeric excess of about 50% with yields up to 90%. So we started to question, why didn't BINOL do the same thing? And what we found was that BINOL is known to planarize in this excited state, and it has about a 50% chance of turning either left or right. And so, even though we started from an enantiopure BINOL, we ended up with a racemic mixture. The work of Tolbert et al. Shows that both BINOL and methylbinol, where only one of these alcohols is protected, both of these return to racemic mixtures within about the same time frame. However, once BINOL was doubly methylated at both alcohols, they did not see any return to a racemic mixture. This shows that the single proton was necessary for the planarization event to occur. Our idea at this point was to take a chiral auxiliary group, attach it to BINOL, and then hopefully from the planar excited state, the chiral auxiliary group would influence the direction that BINOL would rotate, and thus we may be able to achieve some degree of enantiomeric excess. Which brings us to our main paper. One of the reasons that we're excited about this paper is because classically, in order to obtain a higher enantiomeric excess from an atropisomeric compound, you would need to first couple a chiral auxiliary group to your molecule. So here we have BINOL, which then becomes this auxiliary coupled BINOL. You perform either a chromatographic separation or a recrystallization, and that will separate your two diastereomers from each other, usually one of which is just discarded because it's not the desired diastereomer. After which point you perform an auxiliary cleavage, and that gets you your desired enantiomer. However, you got a maximum theoretical yield of 50%. Our methodology is a little bit different in that we're not actually going to be discarding any of our BINOL. We are instead converting one of the enantiomers into the other one. And so we start out with the same auxiliary coupling, but this time we're going to only couple one of those hydroxyl groups to our auxiliary. This is because we need that acidic proton available in order for the ESPT process to occur. After irradiation in the presence of a base, we find that we have converted some or all of one atropisomer into the other one, after which point we can perform an auxiliary cleavage, and now we have a chance to have both the higher enantiomeric excess and a maximum theoretical yield of 100%. So in order to prototype our system, we decided to choose a molecule that was very simple to synthesize and that would serve as a good analog for our system. So we chose the Bach-Pro auxiliary group, so this Bach-protected amino acid proline coupled to our BINOL. We chose Bach-proline as our prototypical amino acid because the addition of the auxiliary group only occurs one time to BINOL. This allowed for an easy synthesis and very convenient workup. We also found that it was very simple to separate the two diastereomers from each other, both the R and the S BINOL, by column chromatography, which then allowed us to study the two different diastereomers and the racemic mixture that we originally generated. Here we see a table of the control experiments that we performed. In our successful system, we have three major components that are necessary. We need to have the chiral auxiliary group coupled to the BINOL, we need to have UV irradiation, and we need to have a sufficient quantity of base in order for the proton transfer event to occur. In our first entry here, we see that with all of the components present, we were able to achieve an 86% yield as well as an enantiomeric excess of 31%. In the next entry down, we did not irradiate our sample, still performed it at room temperature with the base. We got less yield, probably because of the difficulty in workup and separation from the base, and we saw a negligible amount of enantiomeric excess. This was probably just random error. The next sample down, we removed base from our system, and we saw that we had no enantiomeric excess whatsoever. And even without light at a higher temperature, we don't end up getting an enantiomeric excess. The reason that no yield is reported for 4 is because the presence of the base at the elevated temperature actually managed to cleave the Bach-proline group from our BINOL. And finally, to demonstrate that the coupling of the BINOL to the auxiliary group is necessary, we added an equimolar concentration of our amino acid to a solution of racemic BINOL, and we found a negligible change in the enantiomeric excess. So once we had the racemic Bach-Pro BINOL synthesized, we set about trying to get this excited state proton transfer enantioenrichment to occur. By irradiating our sample in the presence of triethylamine, we found that at about 60 minutes there was no longer any change in the enantiomeric excess of our sample. In order to study why this was, we started taking samples at regular time intervals to see how the enantiomeric excess changed. So we found that from 0 to 60 minutes, the enantiomeric excess started changing very rapidly at first, and then sort of leveled off in an asymptotic fashion, which led us to first think about this as being an equilibrium condition. But if this were indeed an equilibrium condition, then regardless of what starting enantiomeric excess we had for our diastereomer, we should be able to come to the same equilibrium enantiomeric excess. And so in order to do that, we performed the same experiment with S Bach-Pro BINOL as well as R Bach-Pro BINOL. And both of these, with fairly small margins of error, showed the same enantiomeric excess, and they both showed that we were achieving the same equilibrium enantiomeric excess regardless of our starting point. In order to confirm this, we decided to use circular dichroism measurements under the same conditions, and we found that they all came to about the same spectrum after the reaction had completed. Once we had found this enantiomeric excess equilibrium condition, we set out to try different chiral auxiliary groups in order to try and find first a higher enantiomeric excess, and also to see if there were any noticeable trends in the shift in enantiomeric excess with the structure of our chiral auxiliary group. The highest enantiomeric excess we found was from our Bach-protected phenylglycine, which had an enantiomeric excess of about 63% under our standard enantioenrichment conditions. While the trend is not very well defined, it seems that the proximity of the chiral center on our auxiliary group to the BINOL has an effect on the enantiomeric excess, as well as the general steric bulk of our chiral auxiliary group. Both of these factors seem to influence the enantiomeric excess of our molecule. In the future, we intend to systematically vary our chiral auxiliary groups in order to test these parameters in a more well-defined fashion.